Thanks very much for the subscription. For those who have subscribed, those who have not yet subscribed, please do one thing: subscribe, put on the notification, and we continue to learn. We continue to enjoy the facts and benefits of studying medicine. Now, today we are going to look at an important signaling pathway, and that is the tyrosine kinase pathway. This pathway consists of monomer receptors which have intracellular protein residues, the tyrosine residues, and it is linked to what we call many effects in the cell, such as cell proliferation and others. So these are some of the proteins we are going to be finding in this pathway. We have the RAF, the MAPK, MYC, mTOR, AKT, P1, PI3K, and RAS. What do they stand for? So when we look at the RAF, this is the rapidly accelerated fibrosarcoma. The MAPK is mitogen activated protein kinase. Then we have the MYC. This is the master transcriptional regulator of cell proliferation. Then mTOR is mammalian target. Of rapamycin. Then AKT is protein kinase B. Then PI3 is phosphorinositide 3-phosphate kinase. Then RAS is what we call the rat sarcoma virus. So these are some of the proteins we are going to be finding in this pathway. Let us go to the pathway and we see what takes place in this pathway. What keep in mind that the hallmark of cancer related to this pathway is what we call self-sufficiency. How does it come? Is due to mutation in different genes. Genes like NF1, that is neurofibromatous 1, which leads to hypertranscription of MYC gene. This is going to lead to hyper cell proliferation and cell cycle progression. Then we have absence of the enzyme, which is supposed to deactivate RAS by converting GTP to GDP. Is also going to lead to continued transcription of MYC. We said MYC is the master transcriptional regulator of cell proliferation. Then let us see what really takes place. So we saw that this pathway has a monomer receptor. But if in the case when we have our ligand binding the receptor, such as growth factor, so when the ligand binds here, we're going to have these two receptors dimerizing. So when they dimerize, we are going to have what we call transphosphorylation at the tyrosine residues. So these are the tyrosine residues. Tyrosine residues. So what is going to occur? We have our bridging protein here. This is the bridging protein. Attached to the bridging protein is our inactive RAS. So this is inactivated or inactive RAS. So inactive RAS is having GDP bound to it. So it is anchored. It's anchored to the cell membrane by the farnesyl membrane anchor. So when we have transportation of the tyrosine residues, we are going to have the phosphate, one phosphate being carried and brought to the inactive RAS, to the GDP on the inactive RAS. So here we're having our phosphate being brought. What is going to happen when you have our phosphate there? We are going to have activation of our RAS. So this is going to be activation of the RAS. So this side we are going to have activated RAS. So this one is activated RAS. So where did the activation comes from? Was phosphorylation of the GDP. So this means that this side we are going to have GDP. So when you want to cut off this pathway, we have to deactivate the GDP. To deactivate the RAS, by conversion of GTP to GDP. So this will be inactivation by phospholipases. Then, if not inactivated, we are going to have the active RAS. So this is active RAS. Active RAS is going to do two things. One, it will activate PI3K. The PI3K is going to activate the AKT. The AKT is going to activate the mTOR, activating our RAF. Then the RAF is going to activate MAPK. Then the MAPK and mTOR, they are going to come in the cell. So what is inside the nucleus? So this is the cell nucleus. This is the nucleus. So what is inside the nucleus? We are going to have our DNA. So we are going to have transcription being switched on. So what is going to be produced by this transcription is the MYC gene. So we say the MYC gene is going to do what? It's going to stimulate one, cell cycle progression by producing of cyclins. Then two, after the cell cycle being activated and stimulated to progress, we are going to have cell proliferation. So the whole of this system is all about cell proliferation and cell cycle progression. So if we have a mutation, for example, in a gene coding for this phospholipase, which is supposed to deactivate the RAS, we are going to have continued transcription and formation of MYC gene, having uncontrolled cell proliferation. Two, when we have, and the gene normally associated with this is the NF1, that is the neurofibromatous one.